All right, welcome back from our break. Our next panel is breast cancer vaccine innovations in the works. And we have three presenters for this panel. Dr. Nora Dissis, the director of the University of Washington's Institute of Translational Health Science and the Center for Translational Medicine and Women's Health at the University of Washington, among other roles. Dr. Kieran Dillon, the Executive Director of the Cancer Vaccine Institute at the University of Washington, and Kristen Dahlgren, who's the founder and CEO of the Cancer Vaccine Coalition. Dr. Dissis, I see you on video. Your slides are up. You have the floor.
Thank you very much. May I ask, is someone else forwarding the slides, or can I share screen?
We will advance your slides for you. Just say next slide for us.
Thanks very much. Thank you so much for the opportunity to talk today. My name's Nora Dissis, and one of my other roles is director of the UW Medicine Cancer Vaccine Institute. Next slide. We're at a tipping point for cancer vaccines, and we're at this tipping point for four major reasons. First, we know the type of immune response we need to have an anti-cancer effect. And that is a type 1 T cell response with cytotoxic T cells and cytokine releasing CD4 T cells that can actually kill tumor cells. The second tipping point is that hundreds of tumor antigens have been identified. And this is really allowing us to create precision vaccines, either targeted to specific tumor types or targeted to the specific patient. The third tipping point is that we have safe and immunologically effective vaccine delivery technologies now. And these are nucleic acid-based technologies, mRNA and DNA, and they allow the application of multi-antigen vaccines, because cancer isn't caused by one protein. And finally, cancer vaccines are being tested in appropriate patient populations, really across the cancer spectrum, from prevention all the way to treatment, being integrated into combination immunotherapy approaches. Next slide. It seems like you can't open a newspaper these days without reading about a successful cancer vaccine trial that's showing promise. And this really is coming on the tail end of a decade of explosion in cancer vaccine research. And this slide really shows that explosion. On the left-hand side of the slide, you can see over the last decade, the number of publications on cancer vaccines really is taking off. And the United States is leading this explosion, with 50% of these publications really coming from our country, and the impact of these publications is tremendous. Next slide. In fact, other countries are taking action based on the impact that our country's research is having on the vaccine field. And in fact, in the last several months, the United Kingdom has started a major cancer vaccine initiative where their plan is to enroll 10,000 cancer patients across a variety of different cancer tumor types and to immunize 10,000 patients with mRNA personalized vaccines for the treatment of their existing cancer. Next slide. Most patients with cancer do have immune responses to their tumors. And those immune responses are to two major categories of proteins. The first category of proteins that are immunogenic in cancer are foreign proteins, and those really comprise viruses such as HPV and cervical cancer, or mutations, which really occur in a certain select group of cancers, such as melanoma, kidney cancer, non-small cell lung cancer. Now, when these immunogens are recognized by the immune system, they give a very strong killer type of an immune response, a type 1 immune response with cytotoxic T cells and really a type 1 immune microenvironment. Although these occur in the minority of cancer patients, this inherent endogenous immune response allows immune checkpoint inhibitors which operate just by unleashing this endogenous immune response as a form of cancer therapy. But the vast majority of cancer patients respond to another category, the other category of immunogenic protein, which are non-mutated, aberrantly expressed proteins. These are more similar to self. They give a self signal. And the immune response that are generated in these patients actually is a regulatory immune response that dampens that type 1 immune response. So when you're constructing a cancer vaccine, the adjuvant, the delivery really will be dependent on the antigen type, because this either will allow you to augment that pre-existing type 1 immune response or have an antigen delivery system, an adjuvant type that will allow you to kind of change that regulatory response into a type 1 immune response. Next slide, please. Now, whether a vaccine is composed of a non-mutated antigen or a mutated or viral antigen, more and more vaccines are being encoded into nucleic acid approaches, either mRNA or DNA. Now, the major reason for these is that these approaches have been around for a long time and they've been shown to be safe and effective. But there are numerous other reasons. These approaches are adaptable to multiple antigens, which is going to be the way that we overcome cancer. These approaches are superior in the generation of cytotoxic T cells in that type 1 immune response. The vaccines themselves are very stable over time and at higher temperatures, so they're easy to travel around. DNA vaccines in particular persist at the injection site for a short period of time after immunization, so they auto-boost the patient. They're low cost and they're rapidly manufactured, and this is really ideal for personalized mRNA vaccines. Next slide. So what I'd like to do now is just give you a couple of examples of these more modern cancer vaccine approaches. And I'm going to start with talking about mutation-based personalized vaccines. Now, many cancers are associated with a lot of mutations, as I mentioned before, and there are different types of mutations. You can have translocations, you can have somatic mutations. These all create unique proteins in the tumor that haven't been recognized by the immune system before. And so basically, to create a mutation-based personalized vaccine, we have to have the patient's cancer. The cancer undergoes sequencing, as you can see on the right-hand side of the slide. The tumor-specific sequences are then analyzed through artificial intelligence to identify mutations that have a high likelihood of interacting with the patient's own MHC or immune system. And then from there, there's an in vitro validation to see if those predicted epitopes actually do interact with the patient's immune system. And then these epitopes are rapidly constructed into a personalized vaccine that may consist of 10, 15, 20 different mutations. So you can see rapid vaccine manufacture is critically important for mutation-based personalized vaccines. Next slide. This is a recent publication of a personalized vaccine for pancreatic cancer. The way the vaccine was made was just as I told you before, taking the patient's tumor at the time of diagnosis. The patients had surgery, so their pancreatic cancer was resectable. The vaccine was made based on the mutations that were present in the patient's tumor. The patients were vaccinated after they were treated with atezolizumab, which is an immune checkpoint inhibitor that helps unleash the vaccinated immune response. Then after vaccination, the patients got standard therapy, which was adjuvant chemotherapy. And then after the chemotherapy, they had booster immunizations. You can see this regimen on the left-hand side of the slide. On the right-hand side of the slide is the clinical outcome. Although it was a small study, the clinical outcome was striking. 50% of the patients were able to develop immune responses against the mutations in their tumors. This can be seen by the yellow bars. And 50% of the patients did not. But those that did immunologically respond to their mutations, compared to those that did not respond, had superior clinical outcomes. Both the relapse-free survival and the overall survival of those patients has not been reached at this time. And this vaccine is now going on to a large randomized phase 2 study. Next slide. Now, artificial intelligence has really advanced the engineering of vaccines targeting non-mutated antigens. What we've found is that within the natural sequence of these non-mutated antigens are epitopes that predominantly elicit those regulatory responses and epitopes that selectively elicit type 1 immune responses. So with this, you can identify proteins that are cancer drivers for particular tumor types. Similar to mutated antigen vaccines, use epitope editing to find those segments that have the right type of immune response, type 1 selective responses, and basically create a multi-antigen epitope-based vaccine very similar to mutated antigen vaccines. Next slide. This is an example of a vaccine designed to prevent breast cancer recurrence, targeting a single antigen, the HER2 protein, which is overexpressed in about 30% of breast cancers. But this antigen is unique as it's a very dominant driver oncogene of this particular subtype of protein. A vaccine encoding Th1 selective epitopes using the technology that I just told you about could be formulated and went into a very large phase 1 clinical trial in very advanced stage HER2 positive breast cancer patients, 66 of them. This was a dose escalation study. The patients had no evidence of disease or bone-only disease, and the outcomes were to look at the safety and clinical response. Next slide, please. The vaccine was very immunogenic. In fact, all doses elicited an immune response. And at the time of when this was reported last year, 50% of patients with stage 3 and stage 4 disease had not had a relapse. And in fact, none of the stage 3 patients had a relapse with a follow-up of over 8 years. 75 to 85% of the patients are still alive. And you would expect the median survival to be about 58 months. And in this trial, still at 10 years, the median survival has not been reached. The vaccine is now in phase 2 randomized studies in breast and gastric cancer and a basket cancer for different types of HER2 positive cancers, such as uterine, lung, and other cancers. Next slide. So this is just a few examples of where cancer vaccines have come quite far in the last decade, and numerous cancer vaccines are moving forward in multiple clinical scenarios. In fact, on clinicaltrials.gov, cancer vaccines is probably one of the most rapidly growing clinical trials that are being today. I've shown you vaccines to treat or prevent cancer recurrence. There are vaccine clinical trials right now to intercept cancer by treating pre-invasive lesions and even to prevent cancer in patients that are genetically high risk. At the Cancer Vaccine Institute at the University of Washington, we have campaigns going on in lung, breast, colon, prostate, ovary, and bladder cancer. These are the cancers that cause 50% of cancer deaths and diagnoses in the United States. And we're working on cancers that are moving through vaccines that are moving through all these clinical scenarios to prevent, intercept, and treat cancers. And with that, I'd like to turn this over to Dr. Karen Dillon.
Thank you very much, Dr. Dillon. You have the floor.
Thank you very much to the committee for giving us the opportunity to share this work. And as you've heard from Dr. Desis, cancer vaccines are here and moving their way through clinical trials. So what I want to focus on for my portion of this presentation is actually specifically talking about clinical applications of these vaccines, which Dr. Desis alluded to. So this includes the prevention of cancer recurrence, treating cancer, and then finally preventing or intercepting cancer. While we're making progress across all of these different areas, the vast majority of the work, our early work, has been in the prevention of cancer recurrence. I'm going to give you examples of work that's happening in each of these spaces that illustrates the progress that's been made. So you just heard about an exciting phase 1 study for HER2-positive breast cancer. And this was published in 2022. But the rationale behind our approach to treating cancer, preventing cancer recurrence, was if we can target cancers at the time where the disease burden is lowest. So giving patients a vaccine once they've had a complete treatment and have no evidence of disease or bone-only disease, it gives the immune system the chance to develop that immune response to recognize and eliminate any cells that might be remaining and subsequently grow. So in addition to the breast cancer study, we've also published a study in advanced ovarian cancer with a vaccine that targets a protein called IGFBP2 that's expressed at very high levels in ovarian cancer. I'm going to talk about the specifics of that study in a little bit when we talk about preventing ovarian cancer. And ongoing, we have 2 phase 1 clinical trials and 2 phase 2 clinical trials, and these are in breast cancer. Now we're moving from preventing recurrence in advanced stage disease more to earlier stage disease, so stage 1 to stage 3 breast cancer. So when we talk about treatment of breast cancer and having vaccines supplement that, a really interesting example is one of a vaccine that we call StemVac. StemVac targets a specific population of cancer cells called cancer stem cells. And this is a small population that actually drives resistance to therapy. They drive metastases, and they drive a recurrence that may happen subsequent to treatment. So we identified 5 proteins that are expressed at high levels in cancers and are found to be associated with cancer stem cells, formulated a 5-antigen DNA vaccine called StemVac. And when we tested this in a mouse model for breast cancer, we found that with the vaccine alone, we could prevent cancer in about 50% of mice. So this vaccine was taken to a phase 1 clinical trial. Again, this is a dose escalation study that was done in advanced stage, so stage 3 and stage 4 breast cancer. These patients had either triple-negative breast cancer or had hormone receptor positive cancer. Patients received 3 vaccinations 1 month apart, followed by 2 boosters at 3 and 9 months. And these patients are in long-term follow-up, but for immune responses, we found that we could detect immune responses to all 5 of the antigens that were part of the vaccine. And boosters were indeed actually really important. They significantly boosted the level of response in these patients, and the levels of responses were quite high. So cancer stem cells aren't just found in breast cancer. They're actually found in many different kinds of cancers, as you can see on the left here, including lung and others noted here. And so we think that this vaccine is actually going to be effective not just for breast cancer, but for treating cancers that tend to have stem cell-like properties, which we can detect using various methods. In fact, we have a phase 2 clinical trial with this vaccine ongoing in non-small cell lung cancer, in addition to a phase 2 in non-metastatic triple-negative breast cancer. We think that StemVac may actually also be really effective at specifically targeting metastases. It's slowing or maybe stopping growth of metastatic lesions. And we're currently developing 2 different trials, both for breast cancer, in one case for metastatic triple-negative breast cancer, and in another for hormone receptor positive cancer. The first one, the triple-negative breast cancer, we hope to open in early 2025. So now moving from treatment to prevention, right? This is sort of where we all want to be, prevent cancer before it becomes invasive. And what you see here is the evolution from normal tissue to invasive cancer all the way on the right. As you can note that there are several precancerous states that can be actually identified in many kinds of cancers. So what if we could develop vaccines that would actually target these precancerous lesions, preventing from those lesions to becoming invasive cancer? And this is in fact what we're trying to do across many different kinds of cancers. And I've listed here some of the precancerous lesions that are associated with various cancers. Prostate intraepithelial neoplasia for prostate cancer, adenomatous polyps. So these are identified during colonoscopy screenings and such for preventing colon cancer. Ductal carcinoma in situ is a precancerous lesion, often identified in breast imaging and that can lead to breast cancer. And it's actually often treated very much like breast cancer. And we think that vaccines will play a significant role there. And then for ovarian cancer, there's a lesion called serous tubal intraepithelial carcinoma. It's a mouthful, we call it STIC for short. And this is actually thought to be to start in the fallopian tubes, and these cells, these STIC cells, migrate out to the ovary or to the peritoneum to seed cancers there. Ovarian cancer is an important one to consider because it's known as a silent killer. It generally is not diagnosed at earlier stages. Usually patients are diagnosed at stage 3 or stage 4. And folks who have family history of ovarian and breast cancer, those who have known genetic mutations, tend to have a higher incidence of STIC that's identified. So these people can undergo preventative surgeries to remove fallopian tubes and ovaries, but that reduces the risk by 80%, but doesn't completely eliminate it. The thought is that the cells have already migrated out of the fallopian tube to seed the cancer elsewhere. STIC can also be found incidentally in people who have surgery for total hysterectomy in a subset of the population. So it's, that population could also benefit from a vaccine. So of course, can we develop a vaccine that specifically targets STIC? And we have evidence from the ovarian cancer study that I mentioned earlier that we think that we can do this. So this is the IGFBP2 vaccine that we tested in advanced stage ovarian cancer patients. IGFBP2 is not only an ovarian cancer antigen, but it's also associated with STIC as well. So we were able to achieve immune responses in patients who were vaccinated. We followed these patients for 8 years and found that a third of these patients didn't recur. And when we look at overall survival, almost half of these patients are alive 8 years after vaccination. For comparison, the median survival for this population is only 18 months. So quite remarkable and something that we're quite excited about. And so we're now developing a vaccine for STIC that not only includes epitopes from IGFBP2, but we're identifying and testing additional antigens to create a multi-antigen vaccine that will first be tested in mice and then hopefully human clinical trials. So as another example of a disease, this is one caused by HPV infection. Vulvar intraepithelial neoplasia is a precancerous lesion that can develop into invasive disease. This study was published by Kincher et al. In 2009 in the New England Journal. And in this case, they vaccinated people who had stage 3 VIN, so this precancerous lesion, with a synthetic peptide for the E6 and E7 proteins of HPV. And then they looked at 3, 12, and 24 months to look for clinical response and immune response in these individuals. And what they found was that at 12 months, 79% of these individuals had some sort of a clinical response, either partial or complete. And 47% of those had a complete response, and that was actually maintained into 24 months. They also saw that all the folks who were vaccinated actually had an immune response, and you could correlate the level of immune response to the actual clinical response as well. So quite encouraging. So the final example I give you for cancer prevention for a vaccine is a case of where we're trying to target a cancer risk factor, in this case, obesity. So obesity is associated with many kinds of cancers, breast cancer, colon cancer, endometrial, others, and of course, associated with all kinds of other disease states, including metabolic syndromes, diabetes, hypertension. And the cause of all of these is thought to be inflammation that comes from fat tissue, so inflammatory fat that can cause a metabolic dysregulation within the fat tissue and also in the immune environment as well. And losing weight does not reduce that problem. The immunologic memory stays even after weight loss has happened. So, so far we've talked about vaccines. Dr. Desis talked about these type 1 inflammatory vaccines. In this case, what we'd have to do is actually reduce inflammation, right? So using the same technology that we talked about, we use the other side of it to develop an anti-inflammatory vaccine by selecting epitopes that would elicit a type 2 anti-inflammatory response. And so that's what we did. We found multiple epitopes that are associated with inflammatory fat, and we developed a vaccine called adipocyte-directed vaccine, or ADVAC. We have very clever names for vaccines. And we vaccinated mice that we feed a Western diet, so high sugar, high fat diet. These mice become obese and they start to display some of the characteristics we see in humans in similar states, meaning a metabolic syndrome. So they become diabetic. And this particular model is also prone to developing breast cancer at high incidence. What you see on the right is when we vaccinate these mice with ADVAC, we can actually correct that diabetes in these mice. This is a glucose tolerance test, but we've also done insulin tolerance tests. And we can prevent breast cancer in about more than half of these mice as well. So currently this vaccine is being investigated very thoroughly to look at impacts on other organ systems and to do extensive safety studies. And we hope to take it to IND in the next few years for human clinical trials. In addition to cancer and diabetes, another application of this might be non-alcoholic fatty liver disease as well. So, so a lot of work is ongoing right now. So with everything that Dr. Deese shared and I shared, I hope that we can all in this room envision a future that's cancer-free, and a large in part to what vaccines can do. And we think about this as vaccines that can really help ameliorate risk for people at different levels of cancer risk. For example, folks who are at intermediate risk due to chronic inflammation. We talked about obesity, but inflammatory bowel disease is another scenario that you could possibly imagine this working in. Those folks can benefit from a vaccine to target that risk factor and to prevent cancer in those individuals. Folks who are at higher risk, so these are folks who might have a genetic mutation or have strong family history, or have had a precancerous lesion identified on a cancer screening, could benefit from some of the vaccines that I shared with you today to intercept that cancer at an early stage or to prevent it. And then those at very high risk who have already had cancer diagnosed and are at high risk either for recurrence or from the cancer itself that's ongoing. I hope you've seen that we have vaccines that are currently in the works to be able to treat those cancers and also prevent their recurrence. So all of this work is done at the cancer, or most of the work I talked about is done at the Cancer Vaccine Institute. We're located in Seattle, and this is our team. And to be able to do everything that I've talked about, we have a very interdisciplinary team that actually has specialized cores, and under one roof, we're able to do discovery work, translational work, and clinical trials. And I invite you to check out our website, UW-CVI, for more information. And I've added Dr. Deese in my emails up there, so please feel free to reach out with any questions. Thank you.
Thank you very much, Dr. Dillon. Our final presenter in this panel is Kristen Dahlgren from the Cancer Vaccine Coalition.
Hi there. Thank you so much for having me. I want everyone to just absorb some of those results for a minute, because those are the results, and that's the conversation that I had with Dr. Deese back in September that made me leave my job as an NBC correspondent to found the Cancer Vaccine Coalition, formerly known as Pink Eraser Project. I've heard this described as the most hopeful time in history for cancer research. And that came from a longtime scientist. We are actually hearing people use the word cure associated with cancer, use prevention associated with cancer. Those are things that we couldn't have imagined just a few years ago. So we all probably have someone who's been impacted by cancer. So just think about that cancer-free future where we don't have to have those toxic treatments, the amputations of a part of our body, where the bulk of the work could actually be done by these vaccines. And that's what we're trying to build at Cancer Vaccine Coalition. So this is me. I'm award-winning, blah, 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 correspondent with NBC News, including the Today Show and Nightly News for more than 2 decades. I was going about my life. I probably spoke with some of you about measles outbreaks or infectious diseases or flu every year. Those were the stories that I was covering. And then in 2019, I was diagnosed with stage 2 invasive ductal carcinoma. I did 8 rounds of ACT chemo, had a double mastectomy, did 25 rounds of radiation. Since then, I've had 6 surgeries, including LVA, deep flap, a revision of the deep flap, flap removal, implants, and more revisions. Today, some of my symptoms, I have no feeling in my chest, abdomen, part of my lower leg. I have limited shoulder mobility. I have lung fibrosis from the radiation. I'm in chemically induced menopause that I have to travel 4 hours every 10 weeks to Memorial Sloan Kettering and then 4 hours back home because I get a shot that isn't covered by my insurance to get it anywhere than at Memorial Sloan Kettering where I started treatment and where somehow they're able to get things like that through. Someone at the NIH at a meeting recently asked me, we have great treatments for breast cancer. Why do we need a vaccine? Well, I give you those examples as why we need a vaccine. Treatment can be devastating. We're called survivors and warriors, but the reality is I will never again feel like my body is my own. I'm constantly aware of what it went through. And I'm not complaining. I'm grateful to be alive, but you won't hear anyone that's a survivor complaining. And the reality is, I would argue that breast cancer has been normalized in spite of some of these facts. So every 2 minutes, someone in the US is diagnosed with breast cancer. 1 in 40 is a woman's chance of dying from breast cancer here in the US in spite of these great treatments. You've probably heard this 1 in 8 figure. That's how many women in the US have a chance of developing breast cancer in her lifetime. And 1 in 3 is how many new female cancers in the US will be from breast cancer every year. So yes, we do see life extending, life-saving treatments for some cancers, but I think we can do better. I was at the Milken conference recently, and I heard a powerful question asked by the founder and chairman of Moderna. He said, why did we put more weight on the lives lost during COVID than we are now? And I think that question is that we saw such amazing collaboration. We saw the pipeline being streamlined. We saw the FDA working to lay out the endpoints, to work with the scientists who were developing COVID vaccines. And so his question is really, can we do that again? And why aren't we doing that again? So in September 2023, I met Dr. Desis, and we were also asking that question. You heard her describe a tipping point for cancer vaccines. So can we accelerate the process? Can we see a COVID-style push to move scientists out of their silos, to engage the public, to involve industry, to streamline the pipeline, and I put this in capitals for all of you, to bring in the government? We have already moved scientists out of their silos. This is our team. So we have Dr. Desis from the University of Washington, Dr. Larry Norton from Memorial Sloan Kettering, Beth Mittendorf from Dana-Farber, Dr. Powell Brown from MD Anderson, and Dr. Tom Budd from the Cleveland Clinic, among those who are working with us. Can we engage the public? Well, that's where a journalist comes in. So when I announced that I was leaving the Today Show, we did get a lot of coverage. We are working on engaging the public every day on spreading the word about cancer vaccines. Yesterday, I just forwarded something about a glioblastoma vaccine that's in development. So we're seeing so much in this space. I would give us a half check maybe for involving industry, but I've also been traveling to medical conferences and the Milken conference, and these are some of the companies that I've been in touch with, some of them who are developing cancer vaccines. And then we get to streamlining the pipeline and engaging government. That is a work in progress. So some of the things we hope to do: we want to accelerate established concepts. We want to quickly enroll studies and design clinical trials to yield swift go, no-go decisions for advancement. And then we'd like to work with the FDA to provide a roadmap a la COVID. We've seen that this can work, and I think together we can do this again. How do I know? Because they're doing it in another country. And while Nora said we are ahead of everything here in the US, I want to offer up the UK, which she also alluded to as an example. So they call it the UK Cancer Vaccine Launchpad. This is a picture of Dr. Leonard Lee of Oxford, who I've been talking with quite frequently. I'm using some of his slides as with his permission and as an example of what can be done. He also invited me to their Great Britain Cancer Vaccine Summit, the first one. They had about 350 people there from all different hospitals and all different cancers working on this. So according to them, a global vaccine technology race is underway. This is sort of where we are. Hundreds of vaccines are in phase 1. About 50 cancer vaccines are in phase 2, and just a few are in phase 3 studies. They've framed this as a race. The UK is winning. I don't know if anyone here is competitive, but they really are saying that they are going to be the first ones to see these widespread cancer vaccine trials. And I would say that we here in America can do something as well. The National Cancer Vaccine Advance, this is when they launched it. So as Nora said, it's this plan to get 10,000 cancer vaccines. This is how it works. So keeping in mind that pharma partners are actually paying and providing the vaccines. So what their government is doing is providing these 16 active cancer vaccine sites, and they're helping to refer and enroll patients. So 500 patients or more have already consented to these trials in year 1. We also have a huge pool of people who really want these vaccines. I talked to organizations like Touch, the Black Breast Cancer Foundation. They have 3,500 women signed up waiting for breast cancer vaccine trials. We just need to get them to them, make sure they're accessible, and get them enrolled. In the UK, these are the cancers that they're currently working on: lung, head and neck, bowel, pancreas, prostate, and melanoma. You'll note that breast cancer isn't yet up there. We already do see some vaccine treatments in bladder cancer, and they're using mostly personalized vaccines, which Dr. Desis went over. Cancer vaccine, thank you. Cancer Vaccine Coalition is vaccine type agnostic. So we want to work with anyone who's developing a cancer vaccine, whether it's personalized or off the shelf, like the ones being developed at the University of Washington. This is just some of the coverage that they've seen. So they really are seeing this widespread public sentiment, public education about the different types of vaccines and the patients that are involved in them. These are just a few of their goals: initiating a global advance of vaccines for cancer and immunity against cancer, transformative benefit achieved across multiple cancer subtypes for early and late stage cancers, and then to place those 10,000 people through clinical trials by 2030. So I'm sure you've all noticed we do not have an NHS. So how does it translate to here in the US? Well, we were ahead of the world or in step on COVID-19 vaccine development. I would say that we have American ingenuity, we do have national pride, we have philanthropy, we have strong industry, we have a history of success in these type of collaborations, from the space race to the Manhattan Project. And we also have a shared hatred of cancer, which isn't nothing. Wouldn't it be nice if we all came together and we saw the country get behind this type of national effort, rallying around something, especially something with the word vaccine in it? I will say I was a little concerned when we first formed that there would be pushback on using vaccine, and I was worried people wouldn't understand that in the beginning, these cancer vaccines would be treatment or therapeutic vaccines. I will say that I haven't seen that type of pushback at all. I've only seen 2 messages on social media, and the worst they said was, "Great, another vaccine." Well, as I pointed out, this is never going to be something that's vaccines are not going to be mandated. Cancer vaccines not going to be mandated by the government. It could be part of your treatment or not. You also don't have to do chemotherapy if that's recommended by your doctor. We have people on both sides of the aisle helping us with this effort. And so it's really encouraging. So what is Cancer Vaccine Coalition? I see us as the hub. So we can bring all these pieces together. The researchers, industry, trial participants, government, we're raising money to help fund these trials. And we're also working with different organizations. I know that the National Breast Cancer Coalition is here as well. And we'd like to bring in all of these groups, all of the stakeholders together to get this moving. CVC creates an ecosystem, bringing together these stakeholders, connecting the researchers to collaborate on the ideas, inspiring each other and innovating. They talk at least once a month together. We're planning a cancer vaccine summit in San Francisco on November 4th. We invite you all. We're also talking, as I said, to industry partners, connecting government stakeholders, philanthropic donors and investors, and connecting patients to enroll those trials quickly. So our mission is to speed the development of vaccines and end breast cancer as a fatal disease with other vaccines to follow. And we're aiming to revolutionize cancer research by being that hub. So instead of what Dr. Dillon and Dr. Desis say could be a long road ahead, vaccines are coming, but in the current system, we may not see them until 2050. Or if we all come together, it's really possible to see vaccines to market by 2030. Did the math, that's 860,000 lives lost to breast cancer in that time. So we could be saving those people. And then think about the impact in places like sub-Saharan Africa, where there's no diagnosis, there's no treatment options. If we were to get these off-the-shelf vaccines, it really could be revolutionizing those places and those women's lives. The reality is we spend $26 billion or more on breast cancer care alone annually. I've heard from these scientists that for less than $2 billion, and I padded that number, we could have safe, effective breast cancer vaccines to market. So why wouldn't we invest in it now? Think about the return on investment, an end to cancer as a fatal disease. Success in breast cancer vaccines also equals success in other solid tumor cancer vaccines. I will leave you with the reason that I'm doing this, a picture of me with my 8-year-old. I want her to have her mom around for years to come. I want her to grow up in a world where she's not seeing her friends getting this disease at an increasing rate, where she doesn't have to either make terrible choices about preventing the disease or live with it and have to go through things like these toxic treatments. I think we can build a better world for the next generation, and I invite you all to join me.
So I want to thank all three of you, the members of our panel, and call for there any questions or comments from members of the committee, either here in the room or online. I'm going to take the chair's prerogative and throw out my first question to begin with. One of our real challenges in infectious disease prevention vaccines is immune senescence. We can have drop-offs in immune responses to vaccine at different ages, depending on the particular vaccine or the particular target. Have you seen any evidence that that's a challenge with cancer vaccines as well?
If I could answer this, this is critically important. In fact, that pancreatic cancer study has gone on to demonstrate that the longevity and persistence of the immune response is associated with those clinical outcomes. Clearly, there will have to be booster immunizations. But in our studies at the Cancer Vaccine Institute, we've seen persistence of memory responses at fairly high levels years after the end of vaccination. Now, in patients with cancer, this might be because they have immunogen in their bodies continuing to stimulate that immune response. And I know those clinical trials that are being performed in patients at earlier stage disease, we have one looking at patients with stage 1 and stage 2 cancers that may not have a reservoir of antigen. Looking to see how long immunity persists is going to be critical, but clearly booster vaccinations will be needed in the long term.
Thank you. Other questions or comments? Well, I think you've stumped us. Fascinating presentations. We want to thank all of you, both for your work. We look forward to seeing more going forward. And Kristen, we like the vision for the future. All right, we have reached our time for the day for public comment. And I understand we have some public comments, both
I think we just have
okay.
one.
One, okay, we have potential public comment in the room. Give our speaker a chance to come to the microphone.
All right, thank you. Well, first of all, I want to just say that was an excellent panel. I have a very strong interest in vaccines, cancer vaccines, and prevention vaccines. So good afternoon. My name is Michelle Tregear. I'm a 15-year breast cancer survivor, and I serve as the Chief Programs Officer for the National Breast Cancer Coalition. And I'm here just to present a little bit about NBCC's perspective and also our work in the breast cancer vaccine space. So founded in 1991, NBCC is a collaboration of activists, survivors, researchers, grassroots and national organizations that we've come together to end breast cancer through the power of action and advocacy. NBCC's major research arm is its Artemis Project, which is an advocate-led, mission-driven model designed to develop strategic and innovative plans to save lives by focusing on primary prevention and the prevention of metastases. Now, when we launched the Artemis Project in 2010, the first question we sought to tackle was, can we develop a breast cancer prevention vaccine? We convened researchers, breast cancer advocates around this bold idea and have since sought to develop a safe, cost-effective prevention vaccine that targets all three major subtypes of breast cancer in order to reduce both the incidence and prevent death from breast cancer. Now, the NBCC and Artemis Project awarded initial seed grants to back in 2011 to investigate potential viral causes of breast cancer, as well as to identify antigen targets for a candidate vaccine, and then also to begin the preclinical work. Over the past 10 years, the Artemis Projects, in collaboration with Dr. Keith Knudsen at the Mayo Clinic, have developed a vaccine construct, as well as the phase 1 safety trial protocol. And in 2020, the Artemis vaccine research plan was accepted by the NCI's PREVENT program, which aims to bring promising cancer prevention interventions through preclinical development. With their support, we are currently in the process of manufacturing the vaccine and anticipate a 2025 phase 1 trial launch. So as our vaccine, as well as these other vaccines, progress through development, clinical trials, and ultimately go to market, we must remember that they must be clinically effective, broadly accessible, and affordable to achieve their goals. Too often, treatments developed via the traditional industry model fail to meet this critical need. That is why NBCC is currently investigating traditional as well as non-traditional drug development strategies to ensure the broadest possible accessibility and affordability. NBCC recommends to this committee that this program establish, if it so chooses to pursue cancer vaccines, a plan to obtain consensus regarding the long-term outcomes that should form the basis of approval of breast cancer prevention vaccine or any cancer prevention vaccine. We also recommend that the program establish a strategic plan to engage broad stakeholder involvement, including patient advocacy organizations, to ensure global access to all cancer vaccines under development that ultimately prove to be efficacious. To achieve optimal public health outcomes, the committee must prioritize making proven interventions accessible and affordable to those in need, regardless of their demographics, geography, or socioeconomic status. Thank you.
Thank you for your comment. Just to let the members of the committee know that there's also written comments from this organization that have been sent to each of you. Are there any comments online? Give us just a moment.
Technical difficulties. Okay, hold on.
All right, we do not have any verbal comments online. As such, we will adjourn our first day of the June 2024 NVAC meeting. I would like to encourage you to join us tomorrow morning for day 2 of our meeting, starting at
9 a.m.
9 a.m. Eastern Time. Thank you for joining us for our June 2024 NVAC meeting. Everyone have a great afternoon.
Produced by the U.S. Department of Health and Human Services.